Hi, Dr. Atchison here, and we're going to talk about brain plasticity and neuroimaging. So brain plasticity is this idea that the brain is plastic. And when you think about plastic, you think about things that can be molded and changed and put in different ways. You can turn plastic into anything, right? So it's that same idea applied to the brain, the idea that the brain can be altered or changed. And there's two main types that we talk about. The first of kind is modifiability. And this is that the functions or the structures can be attuned. Now, this is the kind that your book really talks about that really needs to happen early or have sensitive periods. So this is that, one part of your brain might be meant to do this one thing, but if for some reason it's not getting the stimulation to do that one thing, it'll do something else. Or that there's structures of your brain that are dedicated to language, but the kind of language that you process is going to be different depending on your different environments. So this is, again, the idea that these functions and these structures can be attuned to the environment that the child is in, to the environment the individual is in. And this really has allowed the human brain to be as diverse and complex as it is. The fact that there are so many different kinds of people and think so many different ways and do so many different things and speak so many different languages is a function of brain plasticity. And again, these things are going to need to happen really, really early in development. They're going to have sensitive periods where if it doesn't happen in this period, then it's not going to be able to be changed at all. And we'll talk about that kind of as the course goes on in more detail. So an example of this would be somebody that's born congenitally blind, so they're born not able to see. When you look at the adults who are reading Braille, so they're reading with their fingers, they're reading with their touch, they're seeing activation in their visual cortex, the part of the brain that's done for vision. Now, these people can't see. But the brain's not going to say, ignore that piece of real estate because the eyes don't work or there's something else that's causing vision impairment. The brain's going to say, no, this is still viable brain real estate. We still want to use this, and it will modify it to be able to be used in a different way. The other kind that we'll talk about is compensation. And this is when cells substitute for each other. This is more about recovery. Okay, so if somebody who goes deaf later on, they will see activation in their auditory cortex when they're presented with visual stimuli. Now, it's not going to have that same kind of level, maybe as it's not going to look just like someone that's got a typical hearing or typical vision, but it's, again, this real estate is able to be taken over somewhat. Now, if it's not happening super early in development, then it's not going to be kind of this seamless transition. There will be, it is limited in how this can be done, okay? So its compensation is not unlimited. It depends on how much damage there is or how much function has been lost for some reason. It also depends on the age. So when we were talking about, again, the earlier these things happen, the more plastic the brain is, and also timing and how quickly you can, rehabilitation is started. So stroke patients, one of the most important things is to not only catch a stroke early so they're able to treat it, but to catch a stroke early so that whatever functions have been lost, rehabilitation can start almost immediately because we see that compensation and we see that recovery after these brain injuries is much, much better if we start rehabilitation early. Compensation we can better compensate. Our brain can better compensate if this timing's addressed and it's addressed early. Brain plasticity is affected by a number of different things. Genes will determine how plastic someone, how much modifiability or how much compensation a person's going to be able to have. Experience plays a big role in brain plasticity, especially in that modifiability in terms of what experiences a child is getting, what experiences are going into this brain. I'm going to say this a lot throughout the course. The input matters. So what you input into the system is going to affect the output. So what experiences get put into the brain affects what the brain's able to do, affects the output of that. An example of this comes from a study by Greenborough and his colleagues, and they looked at rats. And what they did is they had two groups of rats initially, and they had one group of rats that was in a non-enriched environment, which is really just a very, very boring rat cage. These were not exciting cages for rats to be in at all, versus an enriched environment. Now, these were souped up rat cages. So this would be like the rat cage on MTV Cribs or something, right? This would be like they would do, if rats did MTV Cribs, it would be in these enriched environments. So lots of tubes, lots of toys, lots of activities, lots of different kinds of things, a really enriched environment. And what they did is afterwards, so they reared these rats in these two different environments. So these were baby rats that were either in the non-enriched environment or the enriched environment. And what they did is they later sacrificed the animals and looked at their brains. And what they found is that there were heavier and thicker cortexes. That means the part of the brain that really does a lot of our function, a lot of the rat's function, had more connections. If it was heavier and thicker, it means it had larger neurons, more dendrites, more synaptic connections. And this was both found for younger and older rats. Now, what's really interesting, and this one doesn't get reported as often, is they did a follow-up study to this, where they compared non-enriched environments versus enriched environments and added a third category where they looked at natural rat environments. And what they found is even over enriched environments, rats that were reared in really natural rat environments that were very similar to what they would be in nature, they actually had even better brains. So, this is important when we think about development. We're always trying to create these enriched environments for children, and we think more is always better. But that's not how the brain works. The brain has evolved to develop in certain kinds of environments. And one of those, the environment that it's developed in is one that has access to things that are in kind of a natural world. So we find that playing outside is really, really important for kids. And this may be one of the reasons. Other things that can affect brain plasticity is brain damage, other deficits, and again, this is where that compensation comes in. Okay, next we're going to talk about, we're going to kind of segue a little bit into function. And what we see is, as we talked about with the neurons, this is basically a circuit, an electric circuit. Because of this, we can measure electrical activity, and what does it indicate? What it indicates is that there is excitability and organization of large amounts of neurons. So areas that have large neurons, neuronal masses, we're going to be able to see that electrical activity in different ways. And this is what's going to segue us into neuroimaging. What's really interesting is when we can take this information about electrical activity and we can compare it to changes in behavior. When somebody's doing something, look at their brain and see where those changes are happening. And this is how we can make those connections between behavior and the brain. So we can measure if we see surges in frontal lobe activity, and we see these, and we see that they're related to inhibition of behavior, planning, goal-directed behavior. So when we talked about the structure of the brain, and we talked about the frontal lobe was doing these things, this is one of the ways that we know that, because we see these behaviors being linked to electrical activity in the frontal lobe. And again, this is due to those synapses and the myelinization being able to sense this electrical activity and being able to it be an area that this is happening. There's different ways that we measure this. We'll talk about several. The main three your book talks about are PET, EEG, and fMRI, and I'll throw one more in there as well. So these different brain imaging techniques, the first of which we're going to talk about is EEG. What EEG does is it measures event-related potentials, ERP. So basically all the E letters go together. And sometimes you'll hear in the literature them talking about them kind of simultaneously. So what you see here is you see the woman wearing an EEG cap. And what you see to the right of her is event-related potentials. That's the information that they're getting. And again, this is that electrical activity. So they're using electrodes to measure this electrical activity. because they're measuring this electrical activity and they're using electrodes to do this, they get really good information about time, how quickly the brain does things. EEG is excellent for that. The thing that it's not great at is spatial information, okay? Because the electrodes on the outside of the skull, it's kind of taking like a core sample of electrical activity. So somewhere in this general vicinity, there's electrical activity happening. And they can make inferences as to where that precisely is, but we're not going to get as great of information, spatial information about where things are happening in the brain. We'll get really good information about how quickly they're happening or the time it takes for them to happen, but we won't get really good information about spatial information. Another one that your book doesn't talk about is NIRS. NIRS is actually kind of using lasers. It's very, very similar to EEG in that it's got probes on the brain, on the skull, and it's shooting in. The difference is instead of measuring electricity, this is measuring light. So it's actually shooting a laser, not a harmful laser, but it's shooting light into the brain. And then it's measuring how much light comes back as a function of how much blood flow. It's going to be similar to EEG in that it's got really great temporal information. It can give us these things really, really quickly, but it's not going to give us good spatial information. As you can see here, we have general idea where these things are happening, but it's not going to be very, very specific. PET measures metabolic activity using radiation. It's going to give us better spatial information, but the problem is it's really, really invasive. You actually have to ingest radioactive materials. Not highly radioactive, not like they're going to hurt you, but it's still more invasive than EEG, NIRS, or the next one we'll talk about as well. But we're getting better spatial information about it because we're able to kind of use this where the radiation is to kind of measure things. The best one that we can do for spatial information is the fMRI. Now, the fMRI is using the exact same technology as the MRI, but they are not the same. So what the fMRI does is it uses the technology of the MRI, but it uses it while the person is active. So that's what the F stands for, the functional. When the person is active, what is happening? So it's using a magnet to record these changes in the brain. It's recording the oxygen levels, and again, that's going to be a function of blood flow and activity. Again, this one has beautiful pictures, gorgeous pictures, but it's got really bad temporal information. So the temporal information, it's not going to give us, we're not going to be able to get quick information about that, which isn't great. And you also have to be very, very still for an MRI, which if you're doing, an infant, it's kind of impossible. Certain demographics really don't like to be in the MRI machines. So again, this kind of very, very still is a con as well because it limits who you can do it with. So we'll see things like NIRS and EEG being used a lot with small children because while you still need to maintain a semblance of being still, it's really just keeping your head still. The rest of your body can have some movement. Whereas, again, with the fMRI, you're not going to be able to have that as much. So again, they have different benefits, and depending on what your research question is, depends on which kind of study might be best for you. There's also differences in price. fMRIs are really, really expensive if you've ever had an MRI taken and you've looked at the bill before insurance. OMG, right? So think about that on a research scale. EEGs, while they're still expensive technology, are much, much cheaper for researchers to be able to engage in. And so again, both of them provide really, really helpful information. So that ends our conversation about neuroplasticity and brain imaging. I'm going to also have a link for a video that you can watch about different kinds of brain activity in different kinds of imaging. So you can see some of these imaging and you can see some of these tasks and what the kind of information they're getting that goes with them. Thanks.